presentar al profesor de biología en la Universidad Estatal de West Virginia. Es profesor adjunto en la Universidad de West Virginia y miembro activo de la comunidad científica, persiguiendo continuamente sus intereses en investigación y contribuyendo al cuerpo de literatura científica. El Dr. Reddy ha hecho y continúa haciendo contribuciones sustanciales al cuerpo de la literatura científica a través de numerosas publicaciones en el área de la genómica, microbioma, selección genómica, expresión génica y compuestos nutracéuticos, y ha realizado contribuciones notables en varios libros. Durante la última década, ha publicado alrededor de 75 artículos en revistas pares como Plant Journal, Journal of Experimental Botany, PLOS ONE, Scientific Reports e International Journals of Molecular Science. Atrajo más de 10 millones de dólares en fondos de agencias federales como NSF, USA, NIFA, NASA y DOT. Damos la bienvenida al Dr. Umesh Reddy.
Thank you very much for all of you coming down here and participating in this training. And my special thanks to Alok for giving me this opportunity. Today's topic is on anthracnose resistance in tomato. So what is this resistance? We need to understand a little bit of basic genetics because the teachers are coming from diverse backgrounds here. In each of our cell, we have a nucleus, and then there are two sets of chromosomes. One set comes from mom, and the other set comes from dad. Okay? And then we have different chromosomes. Like in humans, we have 23 chromosomes. So in tomatoes, we have 12 chromosomes. So 12 comes from mom, 12 comes from dad, totally 24 chromosomes we have in tomato. So inside the chromosomes, this is how it is packed. The DNA is this pointer, the star? Okay. What is it? Oh, okay. Thank you. Let's see. No. Okay. So the DNA, everyone knows this word is pretty popular, and the DNA is quite a bit of DNA is packed in the chromosomes. How that is packed? These bead-like structures are nucleosomes. So the DNA is wrapped around these beads, and then these beads are coils, kind of in the coiled coils, and finally packed in the chromosomes. So they are all tightly packed. When time comes, the entire DNA is not the genes. Only like 5% of the DNA are the genes. Oh, okay, this is excellent. Okay, so this is the DNA that is packed around these beads and coiled into coiled coils and packed into these chromosomes. And when the time comes, that expresses. So the DNA is responsible, the genes are responsible for our phenotype, the way we look, the way we act, similarly for the plants also. And then, we can sequence the DNA, or we can understand how it is wrapped and how that wrapping also is important to understand the phenotypes, and also the genes express. So the expressed genes, all the time, all the sets of genes do not express. Like you are listening to the lecture, whatever is needed for your function currently, that is digesting your breakfast or getting ready for lunch or thinking. And so only certain sets of genes are expressed. So certain genes of the gene, a majority of genes are silent. So when you are subjected to a stress, a set of genes would express. So there are technologies that we can isolate all that RNA and sequence it. So once we sequence it, we call it as transcriptomics because they are the transcribed genes. And then we also can extract from various tissues the metabolomics, the metabolites, and proteins. So we can characterize all these things, how the genes are wrapped and how the genes are organized on the chromosomes, what is the address of the gene, where it is located, and how many alleles are there for a particular gene, and then how these genes express, and how many proteins that cell is producing, and how many metabolites are forming in that particular cell, and all of these are responsible for the phenotypes. So my lab, we work on vegetables, we work on melons, peppers, tomatoes, and watermelons. Especially here, all these crops, especially, all of these very beautiful vegetables that what we buy regularly and use for cooking, and nothing has evolved as it is. Nothing is as it is produced. Okay, everything is evolved from the wild species. The wild species that are completely unedible, we cannot eat them. So that kind of wild plants undergone a process called domestication. So everything, whatever we buy, are the domesticated products. So what is the important process for the domestication is selection. So for 10,000 years of agriculture, these wild plants, the nomadic farmers or the Neolithic farmers, they have selected the plants and then they refined them. And then the modern breeding technologies, they used various crossing procedures to identify the vegetables that are needed for a particular consumer taste, for our particular purpose. Okay, just like you have this Puerto Rican pumpkins, calabajas, which is very specific for your taste that fits in your cuisine. So worldwide, various consumers, they have distinct tastes in their culinary methods, and then they will the chefs and the housewives, they start cooking with various vegetables and fruits in combination to produce recipes, right? So here is the wild pumpkin tomatoes. So the wild tomato, which is Pimpinellifolium, it is very small, tiny tomatoes, and these are, as it is, they are not edible comparatively to the cultivated tomatoes. The tomatoes constitute about $1.16 billion in the United States, and anthracnose is one of the important diseases that is caused by the Coletotrichum species. It's a major disease of several vegetables, not only the Solanaceae family, several fruits and vegetables get affected, especially even causing the post-harvest yield losses. Okay, so how to mitigate them? We need to select pathogen-free seeds. We can fumigate the seeds, or we can do the crop rotation and well-drained soils, lots and lots of fungicide applications to kill the anthracnose-specific disease. So Coletotrichum is a biotrophic fungal pathogen. So in the green plant, the green fruit, if we inoculate the green fruit, then this Appressoria is a specific hyphae that can penetrate. Once it is penetrated, it is going to produce this Haustoria. This Haustoria enters into the cells to suck the juices or the essential nutrients. So this Appressoria will be more or less dormant till it becomes, the fruit becomes red, and then in the Appressoria, in the red fruit, can produce this necrotrophic effect. This is what anthracnose I am talking. If you buy bunch of tomatoes, some of the tomatoes will have this kind of spots. These spots, like after 19 hours post-inoculation, they will develop these kind of dendritic-like structures with swollen hyphae. This is called Appressoria, and producing this necrotrophic hyphae, which symptom looks like this. Okay. So the disease management, the most important thing is applying the fungicides. Coletotrichum is just like people. If we like tomatoes, they also like the fungus, also like the tomatoes. If you kill fungus with fungicides, heavy doses of fungicides, well, the fruit looks good, but that fungicide, systemic fungicides can also harm you, okay, producing cancers or various kinds of diseases in humans. But you may think the fresh tomatoes you have eaten and you had a beautiful salad with dressing, but you are eating a bunch of chemicals as well along with that. So if these chemicals are harming the fungus, keeping the fungus away, it is definitely giving equal effect exactly on your health. That's why what is most important is to identify the genes in the wild species. So all these wild species, while undergoing the domestication, most of the genes were left out. Several of the disease-resistant genes are not there in cultivars. Okay, so we selected the vegetables not for disease resistance. We selected vegetables for these 10,000 years that taste good, that fits in our, our dishes, our cooking. But we didn't consider that what happens to the genes that makes resistance, to build resistance, or they can withstand stress, all that stuff. So we need to cross them back with the wild species. The cultivated species, we need to cross them back with the wild species and identify genes. So not even, no trait is represented by a single gene by as it is. There are so many genes collectively affecting a trait, especially disease resistance is manifested by multiple traits. These traits are located on all over chromosomes, and collectively it is producing resistance. Say a gene is giving 5% effect, another gene is 10% effect, another gene is just 1% effect, another important gene is just 0.5%. So collective action is kind of giving that complete resistance. So we call them, since they are collective several genes, we call them quantitative genes. So each gene is contributing something, so we call them QTL. So we need to cross with wild plants with the domesticated plant or cultivar, and then we have to identify these quantitative traits, that is the multiple genes that are all over the genome, collectively contributing to the resistance. So, and then we need to identify the alleles. For each gene there are alleles. Say eye color in humans is a locus, is a gene that produces eye color. Alleles are like blue color eyes or brown color eyes, black color eyes, different colors of eyes are the alleles of that gene. So for each gene, there are alleles that are producing resistance or susceptibility. Okay, so that is one scenario. One thing is some of the genes are left out in the wild species. They are not even available in the cultivars. We don't have it in cultivar species. So they are the novel genes we need to put back into the domesticated cultivars or the crops, or we need to identify the alleles that are in the wild species that are not there in the cultivars. So we should substitute the alleles with the wild alleles to build up the resistance. Okay, so and then once we identify these genes, if it is a novel gene, we can put all together a new gene using this CRISPR-Cas9 technology. We call it as genome editing. Or if you are identifying a susceptible allele, that is the bad gene, that bad gene can be silenced so that it won't have any effect. So that because there is no bad gene, the cultivar can be resistant. So here is crosses making, say, black is resistant and white is susceptible here. And then you make a cross in the F2 each chromosome will get shuffled, that this has more like a black segment and little bit of white segment, here is little bit of white segment and rest is the black. So you see the different proportions of white and black got mixed up. So the F2 would be further segregating. If you know little bit genetics, the F2 lines are heterozygous and then it would further segregate. So we need to self-it for several generations so that all the chromosomes get fixed. So these fixed chromosomes will not segregate further, and then each we can create like 240, we created for this purpose in the 243 RILs, that is recombinant inbred lines. Each inbred line has its own combination of black and white segments. Okay, so all these 243 RILs, we can screen it with Coletotrichum inoculum. This is Coletotrichum cocodis, which is very important for tomatoes and peppers. So this suspension has 1 mil, has this many conidias we can count, and then we can pipette it out, like, and then this pipette we can insert 2 millimeter depth and inoculate this disease. So after 6 days of inoculation, this fruit lesion starts. That lesion starts. Okay, so this lesion can be measured, and then this data is collected for 2 seasons. So for all these RILs, we need to inoculate systematically for 2 seasons, and then we collected the lesion size data. So the black is totally resistant and white is totally susceptible, that is the cultivar. So here is the wild species and here is the cultivar. The tomato that we buy regularly in the grocery stores looks like this. This is the wild species that is not edible, but it has resistant genes. So to understand where is that resistance, we need to make this cross, and this progeny has to be selfed and fixed so that that will not segregate anywhere and it will remain there. And then 243 RILs were collected, and these 243 RILs were tested for the resistance. That is what we have done here. So, and then we need to sequence all the RILs to understand why it has a certain amount of resistance. And then we also subject another genetic method to identify the genes called genome-wide association studies. So putting together the earlier RILs we have and then the JIVA studies, we can identify the genes that cause the resistance. So there are several softwares and genetic methods to help us to identify these genes. And then these genes, we need to validate them, whether we have identified by mistake or we should apply more than one method to understand, well, these are the genes that are causing the resistance. So another test was done using a bulk sequencing method. I will explain you what is the bulk sequencing method. And also we need to test them how they are expressing, because not just identifying the address is important, and also we need to understand how these genes would express. So that expression data has to be compared between the resistant plants and susceptible plants, how differentially they are expressing. And then once we identify these genes need to be tested in the large populations, whether this resistance and susceptibility remains the way we have understood using these mapping studies. So there are some assay developments have to be done. And then we need to identify the bad genes and silence them, or if there are the good genes in the wild species that are not there in the cultivars, we have technology to transfer the entire gene into the cultivars or the domesticated tomatoes. So here is a susceptible parent that got spread. This is the wild species. You see, after inoculating the disease, the lesion is just there. So the disease is not spreading. Whereas the cultivated one, you see the disease has spread so badly. So if you see these tomatoes, you will just throw it. You think the good tomatoes are good, but they have imbibed a bunch of systemic fungicides. That's why they are showing resistant. Okay, so if these systemic fungicides are bad for the fungus, that is bad for you also. That's why we need to identify genetic means of resistance to build cultivars that can withstand this disease. That is the purpose of our research. So this is how the parents, this is the completely resistant, and this is the completely susceptible. Okay, so and here the distribution you can see in the progeny. A majority of progeny are here towards this one. So what it indicates, the response of 248 progenies we made from this cross are segregating. The extremes are the parents. Some of them even became more resistant than the parents comparatively. Okay, so this is tested for 2 different seasons. And once we sequence these RILs, we need to build genetic maps like this. This is a genetic map. Each linkage group represents a chromosome, and these numbers are there. These numbers are different locus, loci. Okay, so in this loci, we have identified these are showing resistance. Here is one locus, here is one locus, here is one locus. So these are all QTLs. And then what are this green stuff? So now you can see the anthracnose resistance is not single gene. Several chromosomes have harboring genes, and then the collective action of all these genes shows up like resistance. And then these green ones are validated using more than one method. Okay, so more than one method showed that there are very important genes underlying there. And then to understand what are those genes, we need to do further sequencing. So this is one method. The basic method is to identify on the chromosomes what is resistance, what is susceptible. You see each locus, let us say one allele is capital A, and one allele is small a. Okay, so then the capital A allele of that particular locus, for all these 248 RILs, we have 248 progenies, say 50% of them have capital A, 50% of them have small a. Then we need to see the resistance of this, all capital A alleles, all small a alleles. If the resistance is not different, then that particular locus is not linked. Okay, that's that easy. So here what we are seeing as a peak, this peak, that means here is an allele, locus, that has capital A, small a, and then one of the allele is showing resistance, indicating that particular locus is important for the resistance. So that is what we are seeing here, peaks. So all these peaks are they are kind of harboring some kind of resistance. Okay, so but we still do not know what are the genes. We can validate whether those QTLs by doing another study, genome-wide association study, recruiting several other collections, what we do, worldwide collections we can use, and then identify these peaks. So here is a particular locus that has allele that can contribute to resistance. So this is another method. Like here, different colors are different chromosomes, and these parts are single nucleotide polymorphisms. That is nucleotides that are different. Say we all have ATGC. The DNA has ATGCs, the bases. The single nucleotide polymorphism is at a given position on the chromosome here. These two parents are, the cultivars differ. Okay, if some will have capital A kind, some of them will have small a kind. say a particular allele is having GG, that is guanine at a place, whereas the other cultivar has cytosine kind of alleles. So these single nucleotide polymorphisms across all the chromosomes will tell us which one is associated, which one is not, based on the this genome-wide association study of lesion diameter. Lesion is created by anthracnose infection. And also we can do other method to go further to understand what genes are located in these peaks. We made bulks. So 30 progenies that are completely resistant and 30 progenies that are highly susceptible, completely susceptible. So this makes just the lesion diameter of 5 millimeters maximum after infection. These things will make 20 millimeter infection, in the sense lesion size is so large. So that bulks are made for the both seasons, and then we performed deep sequencing, almost like 20x sequencing. If the genome size is, tomato is 750 megabases, so 20x of that sequences, almost like 14,000 megabase kind of sequencing. So this kind of sequencing will tell us clearly what was under those peaks, what kind of genes are responsible for causing resistance or susceptibility. So we can create assays also. We can develop assays of those particular SNPs or loci using this kind of PACE technology. And then that will give this allele discrimination plots. Here is the resistant individuals, here is the susceptible individuals, and here are the heterozygous, and this is control, okay, negative control that is not added in a DNA. So this kind of technologies we use, and then we identified all these important genes. And some of them are causing susceptibility, and some of them are unique genes in the wild species that are coming with resistance totally by themselves. So, and then say here cytochrome P450 is a gene. Here the fourth one, cytochrome P450 in that. That is very important for carotenoid accumulation. The tomatoes look beautiful, red and attractive because of that gene. And that gene causes susceptibility to the disease. Okay, so like this we can identify most important genes that are totally novel, never unheard of, and then they are causing the resistant, and then there are some resistant genes that are already characterized. And these are the allele discrimination plots. Blue is resistant alleles, and red is the susceptible alleles. So this kind of allele discrimination plots can be created. So the CRISPR technology is we can target any gene. So we can create a guide RNA, and the guide RNA, this is called a Cas9 protein that can create double-strand breaks. So that can break the DNA at random places. Say a guide RNA for a gene, a bad gene is can be designed. This one can be incorporated along with the Cas9, and this Cas9 will create double-strand breaks and silence that particular gene. Okay, so the bad genes we can target using this kind of CRISPR-Cas technology and silence them, and totally good genes that are not there in the cultivars, we can altogether catch that particular entire gene, we can sequence it, and we can insert it into the genome. So, and then these are transgene-free technology. There are no selectable markers, there is nothing, it is very safe technology, is about to take over entire agriculture in all crops. Like the first step is identifying the good genes, identifying the bad genes. If the good genes are not there in the cultivars, you can capture those genes from the wild species and put back into the cultivars. In the cultivars, there are bad genes. You can silence them so that they don't have any effect on the fruit quality or whatever, the traits that we are expecting from the vegetables. So this kind of research we do. We characterize the wild species for not only for the disease resistance genes, several important genes for aroma, flavor, taste are not properly selected in the cultivars. Okay? So using this kind of mapping technologies, sequencing technologies, we generate information as well as we identify bad genes and silence them also. So this is our research, like the old traditional QTL mapping and then combination of the modern QTL sequencing approaches like QTL-seq. We combine all these techniques to identify the target genes in tomato. Especially for the anthracnose, we identified a very important gene that is on the chromosome 6. And then those genes can be converted into high throughput assays so that the large number of plants, thousands of plants, can be screened in the laboratory, using very economically, just with a cent, for $1 amount, say about 20 cultivars can be screened just for $1. So that kind of assays can be created. And then the functional approach is this gene knockouts or overexpression approaches will be combined to build the resistance. Rather than chemically treating the vegetables and putting in the grocery stores, very nice, very attractive fruits and vegetables, but they have loads and loads of fungicides and insecticides in them. So you don't want to eat them. So eventually the technology is taking care of building this kind of genomic approaches for crop improvement. That is my research. And then here is Mr. Carlos Lopez Artis. Soon he will be doctor because he did this for his PhD research under my guidance. And then we have two more scientists, plant breeder Padma, and then a bioinformatician. This all is our team. And we have a collaborator at WVU, Wagner, and a USDA scientist. And our funding comes from the NIFA. Thank you all.
¿Alguna preguntita para el doctor?
Yeah, any questions?
Reddy?
Yes, ma'am.
Yes. Sí, tenemos traductor. Alguien que nos pueda ayudar.
Are you considering the possible effect of the modification on the pollinator relationship?
The pollinator relationships, well, tomato is actually supposed to be a self-pollinated crop. But for others, like watermelon is a cross-pollinated. Anything we do, that we need to consider. When a product is developed, say anthracnose resistance, so we need to see the other features, how it has the keeping quality and how it tastes comparatively to the other traditional varieties, stuff like that is important. So what you said is very important. Otherwise, if the flower is changing its pollination behavior, or if it is not producing a nectar, then the pollinators will not come there. So everything is very important.
¿Nos puedes ayudar de traductora a lo que le hace la compañera la pregunta del doctor Umesh? ¿Sí? Sí. Para traducir. Sí.
Buenos días. Este grupo de hongos de la anthracnose creo que es bien típico que afecte muchos cítricos y que afecte otros tipos de frutos, como es el caso de la guayaba. Ante la ausencia, por ejemplo, de la gente
Suave.
que...
Suavecito, porque
Carlos
ella te va a ayudar a traducir.
Yeah, Carlos will
Fabricio.
translate. Please continue.
Fabricio.
Yeah.
Ante la ausencia o la falta de disponibilidad de tener antifungicidas o antimicóticos para plantas de la gente que siembra en la casa. ¿Es efectivo el Malaysian oil, el aceite de malación, para atacar este tipo de hongo?
So Carlos, can you answer this question? Yeah.
Well, what I understood is whether you can use this kind of an oil to treat this fungus at home, right? That's what you're asking? Un tipo de aceite que se usa para matar este hongo en casa.
Sí, la malación.
¿El aceite de qué? Malayon oil. So she's asking about a Malayon oil, whether you can use that to treat this fungus. En
Carlos
guayaba.
can answer this
En
in
guayaba.
Spanish. No, take the mic. Yeah.
Sí, bueno, nosotros no hemos utilizado este tipo de aceite para saber si confiere algún tipo de resistencia en el caso del tomate. Nuestro trabajo simplemente ha sido mayor genético, pero sería una opción interesante poder tratarlo o poder intentar diferentes aceites que podemos obtener de diferentes plantas, como estaba hablando el doctor Héctor, y podemos ver si tienen un efecto antifungicida, pero eso sería para otro estudio.
Very nice.
So, le agradecemos al doctor Umesh, ¿verdad? Su comparecencia en esta semana del taller. Muchas gracias.
Thank you.
Okay.